Cells and organs of the immune system. The many cells, organs, and tissues of the immune system are found throughout the body. They can be classified functionally into two main groups. The primary lymphoid organs provide appropriate microenvironments for the development and maturation of lymphocytes. The secondary lymphoid organs trap antigens generally from nearly tissues or vascular spaces and are sites where mature lymphocytes can interact effectively with the antigen. So, primary lymphoid organs provide appropriate microenvironments for the development and maturation of lymphocytes. The secondary lymphoid organs trap antigen generally from nearby tissues or vascular spaces and are sites where mature lymphocytes can interact effectively with antigen. So, blood vessels and lymphatic system connect these organs, uniting them into a functional whole. Carried within the blood and lymph and populating the lymphoid organs are various white blood cells or leukocytes that participate in the immune response. Of these cells, only the antigen-specific lymphocytes possesses the attributes of diversity, specificity, memory, and self-non-self recognition, the hallmarks of an adaptive immune response. Other leukocytes also play important roles, some as antigen-presenting cells and others participating as effector cells in the elimination of antigen by phagocytosis or the secretion of immune effector molecules. Some leukocytes, especially T lymphocytes, secrete various protein 2 molecules called cytokines. These molecules act as immunoregulatory hormones and play important roles in the coordination and regulation of immune responses. Here we describe the maturation of blood cells and properties of the various immune system cells and the functions of lymphoid organs. First, hematopoiesis. All blood cells arise from a type of cell called the hematopoietic stem cells or HSC. Stem cells are cells that can differentiate into other cell types. They are self-renewing, maintaining their population level by cell division. In humans, hematopoiesis, the formation and development of red and white blood cells, begins in the embryonic yolk sac during the first weeks of development. So, yolk sac stem cells differentiate into primitive erythroid cells that contain embryonic hemoglobin. By the third month of gestation, hematopoietic stem cells have migrated from the yolk sac to the fetal liver and subsequently colonized the spleen. These two organs have major roles in hematopoiesis from the third to the seventh month of gestation. After that, the differentiation of HSCs in the bone marrow becomes the first factor in hematopoiesis, and by birth, there is little or no hematopoiesis in the liver and spleen. The migration of hematopoiesis during development form from a series of early sites to bone marrow is shown. So it is remarkable that every functionally specialized mature blood cell arises from an HSC. However, the study of hematopoietic stem cells is difficult for two reasons. They are scarce, so normally fewer than one HSC per 5 into 10 raised to 4 cells in the bone marrow, and they are difficult to grow in vitro. As a result, the knowledge we have about how their proliferation and differentiation is regulated remains incomplete. By virtue of their capacity for self-renewal, hematopoietic stem cells are maintained at stable levels throughout adult life. However, when there is an increased demand for hematopoiesis, HSCs display an enormous proliferative capacity. This can be demonstrated in mice whose hematopoietic systems have been completely destroyed by a lethal dose of X-rays. When such irradiated mice die within 10 days unless they are infused with normal bone marrow cells from a syngenic, genetically identical mouse. Although a normal mouse has 3 into 10 raised to 8 bone marrow cells, infusion of only 10 raised to 4 to 10 raised to 5 bone marrow cells of the normal amount from a donor is sufficient to completely restore the hematopoietic system, which demonstrates the enormous capacity of HSCs for self-renewal. So early in hematopoiesis, a multipotent stem cell differentiates along one of two pathways, giving rise to either a lymphoid progenitor cell or a myeloid progenitor cell. Progenitor cells have lost the capacity for self-renewal and are committed to a particular cell lineage. So, lymphoid progenitor cells give rise to B, T, and NK natural killer cells. Myeloid stem cells generate progenitors of red blood cells like erythrocytes, many of the various white blood cells, neutrophils, eosinophils, basophils, monocytes, mast cells, dendritic cells, and platelet generating cells called megakaryocytes. In bone marrow, hematopoietic cells and their descendants grow, differentiate, and mature on a mesh like scaffold of stromal cells, which include fat cells, endothelial cells, fibroblasts, and macrophages. Stromal cells influence the differentiation of hematopoietic stem cells by providing a hematopoietic inducing microenvironment (HIM) consisting of cellular matrix and factors that promote growth and differentiation. Many of these hematopoietic growth factors are soluble agents that arrive at their target cells by diffusion, whereas others are membrane bound molecules on the surface of stromal cells that require cell to cell contact between the responding cells and the stromal cells. So, during hematopoiesis, erythrocytes and many different kinds of white blood cells descend from the few hematopoietic cells, stem cells in a complicated process that involves many steps traversing a hierarchy of precursor populations. Why has such a complex process evolved to generate blood cells? Over a lifetime, a person will produce about 10 raised to 16 blood cells. This requires a great many cell divisions. John Dick has pointed out that cell division is error prone and provides an opportunity for the genome to suffer mutations, some of which may cause cancer. He has suggested that to make this potentially catastrophic event less likely, the blood forming system is organized as an indigenous hierarchy in which most of the proliferation takes place within more differentiated precursors rather than in the hematopoietic stem cell population itself. These progressively differentiating precursors are not self renewing, and the mature blood cells they form are either incapable of division or only divide under special circumstances. So, consequently, the chance of generating cancer in HSCs and their immediate descendants is lower, even if it is not reduced to zero. Then, hematopoiesis is regulated at the genetic level. The development of pluripotent hematopoietic stem cells into different cell types require the expression of different sets of lineage, determining a lineage specific genes at appropriate times and in the correct order. The proteins specified by these genes are critical components of regulatory networks that direct the differentiation of the stem cell and its descendants. Much of what we know about the dependence of hematopoiesis on a particular gene comes from studies of mice in which a gene has been inactivated or knocked out by targeted disruption which blocks the production of the protein that it encodes. If mice fail to produce red cells or particular white blood cells and a gene is knocked out, we conclude that the protein specified by the gene is necessary for development of those cells. Knockout technology is one of the most powerful tools available for determining the roles of particular genes in a broad range of processes, and it has made important contributions to the identification of many genes that regulate hematopoiesis. So, although much remains to be learned, targeted disruption and other approaches have identified a number of transcription factors that play important roles in hematopoiesis. Some of these transcription factors affect many different hematopoietic lineages, and others affect only a single lineage, such as the developmental pathway that leads to lymphocytes. One transcription factor that affect multiple lineages is GATA2, a member of a family of transcription factors that recognize the tetranucleotide sequence GATA, a motif found in target genes. A functional GATA2 gene which specifies this transcription factor is essential for the development of the lymphoid, erythroid, and myeloid lineages. As might be expected, animals in which this gene is disrupted die during embryonic development. In contrast to GATA2, other transcription factors, Icaros, are required only for the development of cells of the lymphoid lineage. Although Icarus knockout mice do not produce significant numbers of B, T, and NK cells, their production of erythrocytes, granulocytes, and other cells of the myeloid lineage is unimpaired. So Icarus knockout mice survive embryonic development, but they are severely compromised immunologically and die of infection at an early age. Yet another transcription regulator, BMI1, is a transcriptional repressor that is a key determinant of the ability of HSC to self-renew. And the gene for BMI1, which is highly expressed in HSCs of humans and mice, is knocked out. The BMI1 deficient mice die within 2 months of birth. The cause of death is the eventual failure of the bone marrow to generate red and white blood cells. This bone marrow failure was traced to a lack of self renewal by HSCs. And hematopoietic hemostasis involves many factors. Hematopoiesis is a steady state process in which mature blood cells are produced at the same rate at which they are lost. The principal cause of blood cell loss is aging. So the average erythrocyte has a lifespan of 120 days before it is phagocytosed and digested by macrophages in the spleen. Various white blood cells have lifespans ranging from a day for neutrophils to as long as 20 to 30 years for some T lymphocytes. To maintain steady state levels, the average human being must produce an estimated 3.7 into 10 raised to 11 white blood cells per day. This massive system is regulated by complex mechanisms that affect all of the individual cell types, and ultimately the number of cells in any hematopoietic lineage is set by a balance between the number of cells removed by cell death and the number that arise from division and differentiation. So any one or a combination of regulatory factors can affect rates of cell reproduction and differentiation. These factors can also determine whether a hematopoietic cell is induced to die. Then programmed cell death is an essential homeostatic mechanism. Programmed cell death and induced an ordered process in which the cell actively participates in bringing about its own demise is a critical factor in homeostatic regulation of many types of cell populations, including those of the hematopoietic system. Cells undergoing programmed cell death often exhibit distinctive morphological changes, collectively referred to as apoptosis. These changes include a pronounced decrease in cell volume, modifications of the cytoskeleton and results in membrane blubbing, a condensation of the chromatin, and a degradation of the DNA into fragments. Following this macrophage's morphologic changes, an apoptotic cell sheds tiny membrane-bound apoptotic bodies containing intact organelles. So macrophages phagocytose apoptotic bodies and cells in the advanced stage of apoptosis. This ensures that their intracellular contents, including proteolytic and other lytic enzymes, cationic proteins, and oxidizing molecules, are not released into the surrounding tissue. As a consequence, apoptosis does not include a local inflammatory response. So, apoptosis differs markedly from necrosis, the changes associated with cell death arising from injury. In necrosis, injured cells swell and burst, releasing their contents and possibly triggering a damaging inflammatory response. So each of the leukocytes produced by hematopoiesis has a characteristic lifespan and then dies by programmed cell death. In the adult human, for example, there are about 5 into 10 raised to 10 neutrophils in the circulation. The cells have a lifespan of only a day before programmed cell death is initiated. A stable number of these cells is maintained by constant neutrophil production. So programmed cell death also plays a role in maintaining proper number of hematopoietic progenitor cells for erythrocytes and various types of leukocytes. Beyond hematopoiesis, apoptosis is important in such immunological process as tolerance and killing of target cells by cytotoxic T cells or natural killer cells. So details of the mechanism underlying apoptosis are emerging. The expression of several genes accompanies apoptosis in leukocytes and other cell types. Some of the proteins specified by these genes include apoptosis, others are critical as apoptosis progresses, and still others inhibit apoptosis. For example, apoptosis can be induced in thymocytes by radiation, but only if the protein 53 is P53 is present, many cell deaths are induced by signals from FAS, a molecule present on the surface of many cells. The proteases known as caspase take part in a cascade of reactions that lead to apoptosis. On the other hand, members of the BCL2 B cell lymphoma 2 family of genes BCL2 and BCLXL encode protein products that inhibit apoptosis. So, increasingly, the first member of this gene family to be discovered, BCL2, was found in studies that were concerned not with cell death but with the uncontrolled proliferation of B cells in a type of cancer known as B cell lymphoma. In this case, the BCL2 gene was near the breakpoint of a chromosomal translocation in a human B cell lymphoma. The translocation moved the BCL2 gene into the immunoglobulin heavy chain locus, resulting in transcription of BCL2 along with the immunoglobulin gene, with consequent overproduction of the encoded BCL2 protein by the lymphoma cells. The resulting high levels of BCL2 are thought to help transform lymphoid cells into cancerous lymphoma cells by inhibiting the signals that would normally induce apoptotic cell death. So, BCL2 levels have been found to play an important role in regulating the normal lifespan of various hematopoietic cell lineages, including lymphocytes. A normal adult has about 5 liters of blood with about 200 lymphocytes per millimeter cube, for a total of about 10 raised to 11 circulating lymphocytes. During acute infection, the lymphocyte count increases fourfold or more, producing a circulating lymphocyte level of over 10 raised to 4 into 10 raised to 11. The immune system cannot sustain such a massive increase in cell membranes for an extended period, so the system must respond. In contrast, necrosis, the process that leads to death of injured cells, result in release of cells content, which may induce a local inflammatory response and eliminate unneeded activated lymphocytes. Once the antigenic threat has passed, activated lymphocytes have been found to express lower levels of BCL2 and are therefore more susceptible to the induction of apoptotic death than are naive lymphocytes or memory cells. However, if the lymphocytes continue to be activated by antigen, signals received during activation block the apoptosis signal. As antigen levels subside, the levels of the signals that block apoptosis diminish and the lymphocytes begin to die by apoptosis. And hematopoietic stem cells can be enriched. Weissman and colleagues developed a novel way of enriching the concentration of mouse hematopoietic stem cells, which normally continue less than 0.05% of all bone marrow cells in mice. Their approach relied on the use of bodies specific for molecules known as differentiation antigens, which are expressed only by particular cell types. They exposed bone marrow samples to antibodies that had been labeled with a fluorescent compound and were specific for the differentiation antigen expressed on the surface of mature red and white blood cells, but not on stem cells. The labeled cells were then removed by flow cytometry with a fluorescence activated cell sorter. After each sorting, the remaining cells were assayed to determine the minimum number needed for restoration of hematopoiesis in a lethally irradiated mouse. As the pluripotent stem cells become relatively more numerous in the remaining population, fewer and fewer cells were needed to restore hematopoiesis in the system. So, removal of the hematopoietic cells by selecting for the presence of differentiation antigens allowed a 50 to 200 fold enrichment of pluripotent stem cells. To further enrich the pluripotent stem cells, the remaining cells were incubated with various antibodies raised against cells likely to be in the early stages of hematopoiesis. So, one of these antibodies recognized a differentiation antigen called stem cell antigen 1. So, treatment with this antibody aided capture of undifferentiated stem cells and yielded a preparation so enriched in pluripotent stem cells that an aliquot containing only 30 to 100 cells routinely restored hematopoiesis in a lethally irradiated mouse, where more than 10 raised to 4 non-enriched bone marrow cells were needed for restoration. So, using a refinement of this approach, Nakauchi and his colleagues have devised procedure so effective that in 1 out of 5 lethally irradiated mice, a single hematopoietic cell can restore both myeloid and lymphoid lineages. It has been found that CD34, a marker found on about 1% of hematopoietic cells, although not unique to stem cells, is found on a small population of cells that includes stem cells. The administration of human cell population suitably enriched for CD34 plus cells, the plus indicate that the factor is present on the cell membrane, can reconstitute a patient's entire hematopoietic system. So, a major tool in studies to identify and characterize the human hematopoietic stem cells is the use of SCID, severe combined immunodeficiency mice, as an in vivo assay system for the presence of function of HSCs. SCID mice do not have B and T lymphocytes and are unable to mount adaptive immune response such as those that act in the normal rejection of foreign cells, tissues, and organs. Consequently, these animals do not reject transplanted human cell population containing HSCs or tissues such as thymus and bone marrow. Immunodeficient mice have been useful surrogates or alternative hosts for in vivo studies of human stem cells. So, implanted with fragments of human thymus and bone marrow, SCID mice support the differentiation of human hematopoietic stem cells into mature hematopoietic cells. The system has made possible the study of subpopulations of CD3 plus cells and the effects of human growth factors on the differentiation of various hematopoietic lineages. Then cells of the immune system. Lymphocytes bearing antigen receptors are the central cells of adaptive immunity and are responsible for its signature properties of diversity, specificity, and memory. Important as lymphocytes are, other types of white blood cells also play essential roles in adaptive immunity, presenting antigens, secreting cytokines, and engulfing and destroying microorganisms. So, lymphoid cells, lymphocytes, constitute 20% to 40% of body's white blood cells and 99% of cells in the lymph. There are approximately a trillion, 10 raised to 11, 12, I guess, 10 raised to 12 lymphocytes in the human body. The lymphocytes circulate continuously in the blood and lymph and are capable of migrating into the tissue spaces and lymphoid organs, serving thereby as a bridge between parts of the immune system. The lymphocytes can be broadly subdivided into three major populations: B cells, T cells, and natural killer cells, on the basis of function of cell membrane components. Key cells of adaptive immunity, B cells and T cells, each bear their own distinctive family of antigen receptors. Natural killer cells are large granular lymphocytes referred to their grainy appearance under a microscope. They are part of the innate immune system and do not express the set of surface markers that characterize B or T cells. B and T lymphocytes that have not interacted with the antigen referred to as naive or unprimed are small, motile, non-phagocytic cells that cannot be distinguished from each other morphologically. In their inactivated state, they remain in the G0 phase of the cell cycle, known as small lymphocytes. The cells are only about 6 μm in diameter. Their cytoplasm forms a barely discernible rim around the nucleus. Small lymphocytes have densely packed chromatin, few mitochondria, and a poorly developed endoplasmic reticulum and Golgi apparatus. The new lymphocyte is generally thought to have a short lifespan. Under appropriate conditions, the interaction of small lymphocytes with antigen induces these cells to progress through the cell cycle from G0 to G1 and subsequently into S, G2, and M. As the cell cycle proceeds, lymphocytes enlarge into 15 μm diameter cells called lymphoblasts. These cells have a higher cytoplasm to nucleus ratio and more organellar complexity than small lymphocytes. So, lymphoblasts proliferate and eventually differentiate into effector cells or into memory cells. Effector cells function in various ways to eliminate antigen. The cells have lifespans generally ranging from a few days to a few weeks. So, plasma cells, the antibody secreting effector cells of the B cell lineage, have a characteristic cytoplasm that contains abundant concentric layers of endoplasmic reticulum to support their high rate of protein synthesis and many Golgi vesicles. The effector cells of the T cell lineage include the cytokine secreting T helper cell lineage known as CTL, cytotoxic T lymphocytes. Some of the progeny of B and T lymphoblasts differentiate into memory cells. The persistence of this population of cells is responsible for lifelong immunity to many pathogens. Memory cells look like small lymphocytes but can be distinguished from nerve cells by the presence or absence of certain molecules in their cell membranes. Then, stem cell transplantation holds great promise for the regeneration of diseased, damaged, or defective tissue. Hematopoietic stem cells are already used to restore hematopoietic function, and their use in the clinic is described below. However, rapid advances in stem cell research have raised the possibility that other stem cell types may soon be roughly employed for replacement of a variety of cells and tissues. Two properties of stem cells underlie their utility and promise. They have the capability to give rise to lineages of differentiated cells, and they are self-renewing. Each division of a stem cell creates at least one stem cell. If stem cells are classified according to their descent and development of potential, four levels of stem cells can be recognized: totipotent, pluripotent, multipotent, and unipotent. Totipotent cells can give rise to an entire organism. A fertilized egg, the zygote, is a totipotent cell. In humans, the initial division of the zygote and its descendants produce cells that are also totipotent. In fact, identical twins develop when totipotent cells separate and develop into genetically identical fetus. So, pluripotent cells arise from totipotent cells and give rise to most, but not all, of the cell types necessary for fetal development. For example, human pluripotent stem cells can give rise to all of the cells of the body, but cannot generate a placenta. Further differentiation of pluripotent stem cells leads to the formation of multipotent and unipotent stem cells. Multipotent cells can give rise to only a limited number of cell types, and unipotent cells can generate only the same cell type as themselves. So, pluripotent cells, called embryonic stem cells or simply ES cells, can be isolated from early embryos, and for many years it has been possible to grow mouse ES cells as cell lines on the laboratory. These cells can be induced to generate many different types of cells. Mouse ES cells have been shown to give rise to muscle cells, nerve cells, liver cells, pancreatic cells, and hematopoietic cells. Recent advances have made it possible to grow lines of human pluripotent cells. This is a development of considerable importance to the understanding of human development, and it also has great therapeutic potential. In vitro studies of the factors that determine or influence the development of human pluripotent stem cells along specific developmental paths are provided considerable insight into how cells differentiate into specialized cell types. This research is driven in part by the great potential for using pluripotent stem cells to generate cells and tissues that could replicate diseased or damaged tissue. Success in this endeavor would be a major advance because transplantation medicine now depends entirely on donated organs and tissues, yet the need far exceeds the number of donations and the need is increasing. Success in deriving cells, tissues, and organs from pluripotent stem cells could provide skin replacement for burn patients, heart muscle cells for those with chronic heart disease, pancreatic island cells for patients with diabetes, and neurons for the treatment of Parkinson's disease or Alzheimer's disease. The transplantation of hematopoietic stem cells is an important therapy of patients whose hematopoietic systems must be replaced. There are major applications providing the functional immune system to individuals with a genetically determined immunodeficiency, such as severe combined immunodeficiency, replacing a defective hematopoietic system with a functional one to cure patients with life threatening non malignant genetic disorders in hematopoiesis, sickle cell anemia, and thalassemia, restoring the hematopoietic system of cancer patients after treatment with doses of chemotherapeutic agents and radiation so high as they destroy the system. This high dose regimen can be much more effective at killing tumor cells than therapies using more conventional doses of cytotoxic agents. Stem cell transplantation makes it possible to recover from such drastic treatment. Also, certain cancers, such as some cases of acute myeloid leukemia, can be cured only by destroying the source of the leukemia cells, the patient's own hematopoietic system.